Well, that sponge is beautiful, but yes.
I think, oh gosh, the Picasso sponge. No,
Is that
Star just is coming to mind, but I don't know if that's right.
is that something we need more of a look at, or?
Oh no, 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 no.
Okay.
Just a pretty sponge. Thank you.
So being that it's so dark down here, if we turned our lights off, it would be pitch black. It's really sort of interesting to think about how fish and other mobile organisms get around and what senses they're using to navigate. We've pointed out on some of the fish that the lateral lines is a nice way to sort of orient yourself to your surroundings. I would imagine smell or chemosensory.
Can we zoom in on what's underneath this rock, all those white?
Oh, yeah.
Just curious what they are. I've seen a few, but I'm not. Might just be some worms.
All right, Al, let's zoom in.
It's bryozoans,
Yeah, so
maybe,
not all of that
or
is.
sponges. Looks bryozoans. It looks like it has a pattern to it.
Yeah.
Is this something we want to longer look at, or?
Oh no, no.
Okay.
That was great. Just wanted the confirmation of what we were seeing. It's a mix of those, either those little worms. Yeah, they're probably not anemones. So I'm guessing those are tube worms, and then, like Steve said, patches of bryozoans.
Cool. All right, Al, let's collide.
Thank you.
Yeah, absolutely.
Okay, let's head up slope. We're gonna go 307. Bridge nav. Step 20 meters, 307. Getting sassed by the bridge.
I really need bridge
Sass
channel
from the bridge?
on Sly 1.
Oh yeah, I've been saying please at the end of my last couple of steps, and the last time the bridge said no please, thank you. And then when I called in this time, George said, hello, yes please, Nav. So we're supposed to follow kind of a very brief and short
communication
protocols, yeah, to keep it clear. So step and then the number of meters and then bearing and the bearing to keep it very clear. And there's no room for politeness there. Keep it clear and concise. That wasn't a rock, a C-pen on a rock, was it?
Down at the
I don't
bottom.
think so. Yeah, I don't think it was.
It's on the in between.
Having flashbacks to cup coral.
All right, Al, let's zoom in on this snailfish.
And it was at the end of that
It
dive,
was,
too,
I
that we
know.
were scrambling to try to find it.
We've got 10 minutes, but
There's the
probably a little less with the next watch coming on soon.
nice blacktail snailfish.
One's very pink. Oh, look at his little eyes looking at his little world around him. So big. He's so small. Oh, that's a good shot under his chin. You can see his little pelvic fin that's been altered into like a
Mhm.
little claw where he can stick onto those tanner crabs.
I wonder if it uses it too to sort of
All right,
disturb the sediment.
let's come on. I do want to.
Nice shot though.
Yeah.
You know, in Milton Love's book, it talked about them being preyed on by stellar sea lions, the black-tailed snailfish. And I was just thinking, we don't really see stellar sea lions down at the Channel Islands National Marine Sanctuary anymore. I know it's part of their historic range, but it seems like they mainly are found north of here now. Every once in a while, we'll get sort of a wayward male. There was one that was at the entrance to Ventura Harbor a few years back that was on the whistle buoy with a bunch of California sea lions. But they're very rare to see around here anymore. Looking at these white sponges, Lizzie, just to make sure that we're keeping an eye out for the ones that Abby's interested in still.
Do we want to zoom on these?
I think let's see, the one on the right.
Yeah, that one looks more
Looks good.
interesting. We've seen more of these barrels and we have this frilly one, so maybe we can
Let's-
zoom in
Mhm.
on this
Let's get
quickly.
a quick zoom. If we don't have long, but
Actually, the ship has
Yeah,
just completed a
that's okay.
move, so
Oh.
we're good.
Let
As
me
long
look
as
at
you want.
her.
Oh, that's an interesting one.
sorry, I think my back end is kind of near something. It's gonna be Kamad.
That's okay. I don't see this being one of those
I don't
images
see it on
she
her
sent us,
images.
Al, can we
Mm.
come
but
on?
it is interesting for a zoom.
It's
'cause
something
maybe she'll
different that
put
we
it on
haven't
her
really
list.
seen. Yeah.
Is it something we need a sample of, or good to, or a better zoom of?
No, I think we should keep going.
Okay.
Maybe a better zoom if we have time to do it quickly. But if we have to stop the ship, I guess you just said it stopped, right?
Ship's already holding. We just completed a move,
Got it.
so good timing.
Got it. Okay, yeah, let's do a good zoom, and we'll get a good picture. And if she needs to put this on her list for future collections, then she can have those beauty shots. It is really
Mm-hmm.
Got it on the lasers. Can we go type for her? Can we do a no laser shot?
Yeah.
Any closer?
I'll need a tilt up.
Yeah.
I can go closer, but
Yeah, you can go close. All right. Is that good
That is
size?
interesting. Yep, that is great.
Yep.
Thank you.
All right, let's
Thank
come on.
you.
Oh,
That crab is
Oh.
booking it.
Oh,
that's
Oh,
a lot of
wow.
crabs.
slowly descending. It's kind of like one of those where you're stumbling, but you're still on your feet because you're just
So
trying
we've
not to
got
fall
two.
on
We've
your
got the
face.
king crab. Is that a king crab? And then we've also got the tanner crab.
Mm-hmm.
We're drawing a crowd.
This
All right, we
is
can keep
apparently
going
crab
along.
territory.
Lasers
We are
back
not
on.
welcome
So
here.
where's that? We need the, now if we just had the blacktail, yeah. Now we need the blacktail snailfish to come in and attach to the tanner crab. and then
Oh,
our
I would pay
Alright.
our
so
view will
much
be
money to see
complete.
that.
So
What's our next?
we have 6 minutes left, and I suspect the ROV team will be here in just a couple. So I think this is a good time to
Yeah, we
grab
can hang
another
out.
quick couple zooms if you
Oh,
want.
Okay.
Yeah, let's find something interesting.
To
okay.
the left, there was a yellow sponge. I don't know if that was of interest. A little farther left.
A lot of stuff on this rock up too. Boulder up top too. Can we put lasers back on?
Yeah,
You want to do a boulder?
yeah, unless that sponge is
Yeah,
exciting
let's look at the
anyone.
boulder.
That's a good place to stop too to hand over, I think.
Yeah, I like being out in front a little bit better, the handover too.
Another one of those possibly long spine thorny heads right there.
Yeah, short spines tend to be shallower, so I'm wondering if we're getting into short
You know,
spine
drop down under
territory.
the boulder.
Pardon?
Oh, then they
I think we were
definitely need
trying to
to
look at
flare
the
their
boulder
dorsal
itself.
fins so we can
Yeah.
take a better
Okay.
look.
Mm-hmm. Agreed.
Look for that dorsal spine length.
Ah, gotcha.
Okay, let's zoom in. I'll drive forward just to get it in frame.
It's hard to say.
Flare your dorsal fin. We need to see.
I know, right? Spook 'em.
Alright, let's come on.
I think it's a long spine. Let me look at
Yeah.
I'm gonna compare between the two, but I think we're still looking at. Let me find the other.
What is that
Yeah,
little
I think
white?
that's a long spine. You were seeing sort of there was some white pigmentation on the dorsal spines, and that's definitely the long spine. You can see that the short spine one doesn't have as much of the white pigment with the spines from the images that I'm looking at in Milton Love's book.
Got it.
Okay,
I got it.
yeah, I think we're just about ready to do the handover over here.
Okay.
Awesome.
It's been good working
So
with you
we're
guys
about
this
to
morning.
do. Oh
Alright.
yeah, yeah. Thanks for everything, guys.
Yeah, take care.
You too.
Have a good break.
And who takes over for
So
us?
we're gonna. Yeah. Meredith. And is it? Gotta pull up my schedule. I think today's Friday, so I feel like there's a couple swaps. I'm gonna say Kurt.
Ah.
Meredith and Kurt Whitmire. So we're gonna do a watch shift change now. Julie and I have the pleasure of narrating your dive experience today from 8 to noon. And we'll be back on again from 8 p.m. to 12 a.m. tonight because this is a 32-hour dive. So we have quite some time to explore this region. And thanks, Julie.
Thanks, Lizzie. It's been a lot of fun. I've been learning some new things. Every time I dive with you, I learn some new things.
Oh, yeah. Yeah, it's been and having Laura come in and moderate public questions is also great.
Yes, that was fun. All right, well, I will see you or hear you, log back in with you at 8 o'clock tonight.
Yes, that sounds great. Hear you then.
Okay,
Bye.
bye.
Hey, this is Kurt doing a sound check.
Hey, Kurt, you sound good.
Great, thanks.
Hi, Kurt.
Hey, Meredith, how are you doing?
I'm good. I'm just quickly muting cameras here.
Yep, I'm getting all set up myself.
Oh, I just wanted to
Hoping to
give you a little advanced warning. Up here, we're getting a little wind that's supposed to be escalating through the afternoon. It's not supposed to be anything significant, but if I suddenly vanish on you for a few minutes, it's possible I've lost power.
Oh, wow, okay.
Yeah, I've got a
Good to know.
hotspot, so I'll be able to reconnect, but I just means I'll have to go fish out the hotspot.
Roger that. Just getting up to speed. I was on a call right before this, so I couldn't listen in, but it seems like have we done 5 transects so far?
Yeah, 4 or 5. And I just talked with Lizzie on the Nautilus channel. We are headed towards waypoint X right now. We're almost there. They had a long kind of pause while they sampled some urchins and then sorted out some stuck urchins. So if we could make more tracks towards X and then Z, and get some more transects in, she said that would be good. Which I don't think will be a problem. I don't know how much, given what we're looking at right now, we'll stop to collect. And
Roger that.
Hello, everybody.
Good morning.
hello.
Morning.
Good afternoon, I guess.
Oh, yeah, it is afternoon for a few minutes.
So just here and taking a look at HiPAC, it seems like we want to get over to X and Y and probably along these steeper slopes as we go.
Yeah, if we could stay on the steeper slopes as we go, that would be good.
All right, so we'll just go that way towards X, and then we'll hop up to this ridge over here on our way to Y.
Yeah, that sounds great.
Okay. Ready to move on?
Alrighty.
Yep.
Okay. All right. I think we'll be roughly start out going 295, see what happens along this.
We're gonna go up the slope or along it?
We want to go along it, and Argus is facing. Let's see, 295. The general trend is 295, and we may have a little bit of curving.
Roger.
Start with that. Shallow enough. Bridge now. Actually, yeah. Let's go 30 meters, 295.
It's a very steep slope, but not much consolidated substrate.
Mm. What kind of coral species would we expect to see at this depth?
Here we could see some of the Paragorgia of 2 different possible varieties. One that's the dark pink we've seen in some other locations, and the other is the lighter Stephen Cairnsey, what the Southwest Center calls the peppermint coral. We can see a lot of the Plexarid gorgonians at this depth. We're seeing just above that grenadier is an Anthoptilum sea pen. We've been checking those to see if any of them are attached to rocks. So far, they're all been in the mud. Could see some Plumerella.
Zoom in, Steve.
These always look so much nicer underwater than they look once you get them as a sample.
Mm.
Yeah, it's nice. All the polyps extended. So it looks like so far in this dive, we've collected some sponges and we have pink urchin for
Okay.
Taylor Ann. 3 of them. Or no.
Yep.
3 in box 2 and or 3 in box 2 and slurp jar, so 5 of them.
Yes. There was some excitement
That's
with one getting, they were trying to do the vacuum pickup and it went a little past the lip of the hose and then they had an adventure getting it to come all the way through the hose.
I bet.
Did they wind up getting it
That's
through?
great.
It sounds like they did.
And it looks like we got a Niskin at the end of transect 2 and a couple push cores as well a little later in the dive.
Can you look at the craft arm and see where it is? The bubble camera? Yeah, good. Cheers. Thanks. Look like it's down a little.
For anyone just joining us, this dive is happening on the Footprint Reef in the Channel Islands National Marine Sanctuary. This is an area where some of the shallow areas have been pretty well studied, but there's been very little work done in the deeper areas of this reef. So we're starting
Zoom in
down
there,
deep
Steve.
just to survey the deeper portions of the reef.
Thanks.
Nice tube sponge there.
Bridge
The
nav.
variety that was collected for
At
Abby
50
this morning.
meters, 295. Thank you.
Okay. Oh, come on. Auto heading for you. Had auto heading on and it got caught on a rock and kicked the other vert or the other horizontal.
Which was the one that's giving you trouble?
It's the Argus.
Oh,
Pressure.
Argus. Got it.
You want to stop and look at these sea pens or?
I was actually gonna say if the ship is moving along, why don't we go ahead and just start off with the transect?
Sure.
Can do. Let me know when you're ready.
Yeah, I'm ready.
Okay. Which one's that, Taylor Ann?
Let's see.
6?
6, yeah.
Nice little bristle mouth there.
A few different sponges, a beautiful white base sponge here.
I like the tube on the end of that one.
Yeah,
Looks like
nice.
a funnel.
It's like a caliphate crab down there. We did see a type of king crab earlier. I think that's the Paralomus barreli they've seen occasionally.
I seem to have been seeing a ton of snailfish in the last hour.
I did hear that, yes.
Nice hairy barrel there. There's a crab. So have we started the transect officially?
Yes.
Okay.
Oh, yeah. Here, let me
Oh, I see down in the notes. That's all right.
Yeah, I can take care of that, Meredith.
Okay.
There's the transect tracking.
Just waiting for it to go by in the science chat and not looking at the sample log down below.
Bridge now. Add 200 meters, 295. Thank you.
So Kurt, have you done any of the previous work that was up on the top of the reef here?
I have been to Footprint Bank a couple times, and we've been here for a couple different projects, one associated with West Coast Deep Sea Coral Initiative, and then another associated with the Fisheries Untrawlable Habitat Strategic Initiative, where we were essentially trying to understand fish behavior in response to mobile survey platforms like ROVs and human-occupied submersibles and AUVs. So we were up at shallower depths on top of this reef, kind of on the order of 120 to 130 meters along the, I guess, the spine of the Footprint Reef. So this is my first time actually watching a live dive on some of the deeper flanks of the reef. So it'll be nice to kind of come up slope and merge with some of the habitats I'm more familiar with.
Nice.
Yeah, and you can see that, you know, we have a lot of essentially consolidated rubble that it probably eroded off that some of the top bedrock features of the reef and tumbled down this slope on the southwestern side. You have some sediment, looks like some silt sediment drape over a lot of these cobbles and boulders.
Yep. Yeah, I started watching early when Tom started his shift, and they were in really big boulders down deep, and then it would kind of alternate big boulders and then flat sediment, and then big boulders and then flat sediment. And in the last little while, it seems to have been mostly this kind of cobble slopes.
Yeah. I suspect, yeah, we'll see more of those patterns moving up. Some of the steeper sections, we should get some more exposed outcrops, hopefully, and then additional areas like this.
It's interesting that with all of this rock, you would think there would be a little more coral or sponge, but there must be something right in here that's not exactly optimal for them, whether it's the high sedimentation or something about the flow rate.
Another crab, a different crab, but looks like a lithoid crab of some type.
Some thorny heads all tucked in to the rocks. think that's a big dead sponge up there. I don't know if it rolled
Oh,
down
yeah.
from above or they were here and now they're not. So while we're on the transect, we could do a quick round of introductions. I can start. This is Meredith Everett. I'm from the Northwest Fisheries Science Center with NOAA. I'm a geneticist and I work with the Deep Sea Coral Research and Technology Program, doing the genetics and taxonomy on deep sea corals and increasingly some sponges, and the environmental DNA sampling associated with these communities. Kurt.
Yeah, and I'm Kurt Whitmire, and I'm also at the Northwest Fisheries Science Center with Meredith, and have worked at the West Coast for the West Coast Deep Sea Coral Initiative and the larger NOAA Deep Sea Coral Research and Technology Program for, gosh, over a decade now. And I do a variety of habitat research and analysis of data, and am most interested in how deep sea corals and sponges relate to what's called essential fish habitat, and how we can protect these habitats and minimize the adverse impacts of these habitats from various activities, but namely fishing. So it's essentially working to try to understand the importance of these habitats and conserving habitats while also maintaining fishing opportunities in other areas that have lower impact.
In the van here, this is Stephen Matter on video engineer. Jake Bonney in the Argus chair.
Dan operating Hercules.
This is Renato Kane in the navigator seat.
And this is Taylor Ann, back here logging all the data.
And nice to meet all of you virtually. I think this is the first watch I've had with this watch on the ship, so
Yeah, I think
it's
so.
a pleasure.
Glad to have you.
Yeah, we were supposed to do one of the middle of the night watches together, but as is typical with field work, the calendar and watch schedule has been a bit of a moving target.
Yes, in these type of operations, we need to stay as flexible as possible.
I'm going to change course to 310.
Roger.
Bridge nav. Change course to 310. Thank you.
Are those the Icona clams that they've been seeing previously on this dive?
With the orange lip?
Yeah.
I think so.
Definitely into some siltier section of the habitat here, so we're seeing a lot more of these fragile urchins, and then the feather boa sea pens, the Anthoptilum. I believe a little while ago we did see a cat shark.
Mary Wixton ashore, who's been following on and off through the morning, identified that crab, the really spiny one, as a big spiny lithoid, is a Paralithoides californiensis, always light color, never dark red.
Thanks, Mary, for that help.
Got a big fin eel pout down there.
Oh yeah, a little grenadier.
Oh, and a few more sponges. Oh, that's a nice one.
Oh wow, yeah.
Perfect.
Any chance we can get a quick
More,
zoom on that?
On transect.
Oh, sorry. Yeah, I forgot we're on transect. Never mind.
Gotcha.
Thanks, Rennie, for keeping me honest.
Yeah.
And Steve.
No worries.
I would have had to tell Tom. It was Kurt's fault.
Yeah.
Well, they will have stuff to count. It'll just largely be sea pens and thorny heads. And I'm guessing I see a little fish tucked under the rock that's just to the right of the lasers, and I'm guessing that's Tom's favorite ruby-nosed brochula.
He does love that one. He's got a lot of favorites.
Yes.
It's good. Look at that. Looks like a squat lobster. Look at the length of those forward
The arms.
appendages. Yeah.
They're pretty long.
Yeah.
Mushroom coral.
It's like the crab has its own manipulator arm.
Thorny head. Tipple. And
Is this the
should
depth
probably
where
change
the
my
short
view.
spines and the long spines can overlap?
Yes. And I believe at these depths, Tom has reminded us that the larger ones are typically short spine. And then as you come up in depth, you'll get into more of the long spine.
Do you want me to follow
Short
you up
spines
there?
are, I think, deeper,
Yeah.
but they have
I don't
overlapping
know where we
distribution.
need to go to stay on the steep stuff.
Yeah, the steep stuff's up here, so we're going in that general direction.
Alright.
I'll change to 330.
Roger.
Bridge now.
Bring your head to the right some, Jake.
Change course to 330.
I see. Moving back up the hill to where the slope is.
Yeah,
Yeah.
just inching our way over there. Let's check out that feature, slopey feature.
I didn't realize I dogleg so hard there. It's following the sonar.
Yeah.
Although the big barrel sponges seem to like the flat.
Looks like you could probably go north for a while.
Roger. Bridge nav. Change course to north. Thank you.
Long worm tube there. Just going below the lasers. Sometime I want to get out with a low light camera and poke at these Anthoptilum until one of them bioluminesces.
There's a large crab there off to the upper right. I don't know if that's a parallelus or not. Kind of looks like it. Maybe
Mary
Mary will
says that
chime in.
the long-armed crab we saw was Munida hispida. She said it's the only local species that has long arms. And it's a full adult. The smaller versions don't have the proportionally long arms yet.
Okay. Yeah, Mary, it seems like all these sea pens are after their polyps fully out.
Yep. All basking in whatever current there is here, feeding away. That's one of the ones that when we get it from the lab, it's clearly very slimy on shore or on deck.
Yes,
It's
and
also
you're
an
right that it
interesting
doesn't
one.
look.
Go ahead.
I was just saying, you're right. They don't look quite as nice when we have them on deck as opposed to what we're seeing now in the in situ photos.
Yeah, that species is actually interesting because it's super common, and we were trying to get it into our genetic voucher library, which is a genetic library of sequences for identifying the species that we use for the eDNA and all of that. And for the longest time, we could never get good sequence out of it. We couldn't figure out why. And it turns out that it is one of a handful of sea pens that has a mitochondrial genome rearrangement. And its mitochondrial genome has the same order as the bamboo corals.
Mm.
Oh, wow.
So the primers we were using to amplify it just wouldn't work because they cross a gene-gene boundary.
What's that?
Nice.
Another sponge
Dead
right
header
there in the
cone.
middle, heterocone. Quite a bit of sediment on that one. It's still hanging up. Actually, it's partially toppled over, isn't it?
Yeah. Maybe it just attached to a rock that was not quite big enough.
Yeah, they overgrow their rocks sometimes.
There's another one of those Munida crabs just going off the screen.
to 20 altitude. Roger.
On it.
Yep, coming down.
I like the way the barrel sponges just suddenly loom out of the darkness ahead. Do they blend it in because they're the same color as the sediment on the outside?
Yeah, and that wasn't a super large rock that it was attached to either. Looks like we're getting some bigger chunks of bedrock that have broken off.
Yeah, we've been coming up the hill for a while.
It did look like a hake, yeah. And there's another one of those squat lobsters, the Munida species, or that's the genus. For some reason, I'm having trouble pronouncing that.
I'm going to alter back to 295 so we get over there.
Roger.
Another eopalp, bottom
Great
screen.
snap.
And another grenadier.
These
I feel
are
like we saw that pair earlier.
Yeah, different than we
Change
saw
course
yesterday.
to 295. Thank you.
Species must be shallower. And another cat shark.
A couple of bigger boulders here.
Are we up on top now? Or
This is a little bit of a ledge here, so we got into the flatter bit, but still going up. I want to go this way to hit that steeper slope.
copy.
That sponge found the very apex of that rock
Yeah,
to settle on.
perched right on the very edge of. It almost looks like it
And that
might be growing on like a dead sponge, another dead sponge.
Yeah, maybe that was it. I should probably close my blinds here. I'll see better.
The perils of broadcasting from home. You don't have the nice, perfectly pitch black van with the enormous wall-size screens in front of us.
Yeah, I was trying to get a third monitor in my setup right before noon, but it just didn't work out. For some reason, my laptop isn't recognizing it.
I've been operating the whole season on 2 laptops. One with the science chat open and one with the video.
Well, Jen Brown, who's been on watch over the last month or so, occasionally has sent me a text of her setup, and she had, I think, 2 laptops and 4 monitors. So she had gotten, I guess, taken laptops from all of her family and extra monitors and had a nice setup. I was pretty jealous.
Yeah, I tried to do some of this from my living room where I can put the video up on our giant television screen, but we've had construction going on in our yard for the last week or so, and that just led to a lot of noise. There's the short spine thorny head.
Yep.
All right, we can call that the end of transect.
Okay. All right.
All right.
So steered towards the slightly steeper slope that we'll be approaching, hopefully. See what it ends up looking like.
Sounds great, Rennie.
Saw that guy in our tail camera.
Yeah, you did a
Oh,
good
nice.
job steering around it
Oh, wow.
for as tall as it is.
Yeah, I thought I was
Zoom on that.
afraid I was going to hit it. Yeah, zoom in there, Steve.
Well, that rock hasn't moved for a while because that sponge is pretty large.
This is, I think this is what gets called heterocone, but is likely a different species than the other ones we see.
Can I grab a shot without the lasers?
Sure.
Yeah.
Pull up the DSC over there, Jake.
Hold up. Do you want me to hold up here?
Come on, let's get a quick shot of it.
Yeah.
Yeah, you might as well stop him up. Let me get back out in front.
Bridge now. Hold position.
So Mary Wixton on the chat counted or instructed us to count the walking legs behind the pinchers, and if there's only 3 sets of walking legs, it's a lithoted crab. If it's 4, it would be a tanner crab, a groove tanner crab, which is the only tanner crab species in Southern California bite. Thanks for that information, Mary.
Got a little swiftia coil
Although
down
this
at the
is,
base of that sponge too.
oh yeah. Enter this transect information here.
What kind of crab is this?
I don't know. I'm waiting for Mary to get back to me. I'm not confident on my crab IDs.
Yeah, I got out my crab ID book here, but it's for areas that are much further north. It's for Pacific Northwest and typically shallow species.
Hello, doggy.
The wind is blowing apples off our apple tree, and one just hit the side of the house, and he thought someone was knocking.
Oh, you weren't kidding about that wind, huh?
Yeah, really.
No, it's quite windy. And I live right on the crest of a hill with a big field, so it's got a nice long fetch before it gets to my house.
Mm.
Nice.
I like all the little crabs tucked in there.
Yeah, those are good spots. Get up high. Wait for the sponge to
Little fish in
get
there
caught
too.
in the folds.
Yep.
Oh,
A
yeah.
little isoline sculpin.
Zoom in on the sculpin there, Steve.
Gladly.
Thanks. Got him a nice little
Oh,
home.
he's very cute.
It's a great shot.
Good shots on the DSC.
Yeah. Came most of the way around it. Or 3/4 until I hit that rock there.
Erica Burton is asking how tall we think this sponge might be.
I can put the lasers back on for scale.
Yeah, when we zoom back out.
2 meters, I would say.
I mean, wide.
For the height of the sponge?
Mm-hmm.
Yeah.
Yeah. Say close to 2 meters.
Wow.
Yeah.
So
Impressive
get a
sponge.
good shot of it there in Argus.
Do a zoom in Argus. That's all right.
Sure.
I'm blocking it now, but
It's fun to think about a sponge that's taller than I am.
yeah.
It's a good
I
shot.
get a real good sense if I sit down here on the seabed.
That's a great shot in Argus.
Yeah, a good sense of scale with next to Herc.
That's Herc's bumper there.
That's crazy.
Probably put the DSC into the thing. Get a close-up of it there with the stealth camera.
I'm looking forward to seeing these still images when the data comes back.
You'll lose the light there.
Someone else is too.
Oh, it looks awesome. And the stills camera.
Just took a shot.
Nice.
Nice.
Try one more and get it centered up there.
Yeah.
Get the lasers off.
Oh, yeah.
Can you even?
Nice shot there.
Yeah, we're trying to get a picture of the crab with our digital still camera. That's
Ah.
It's more downward looking, so it's looking like straight down the sponge.
Awesome.
did it shoot?
Yeah, it shot.
Sweet. Yeah, it's the stills camera right there next to bubble camera. Pointed right down in
That's
the
where
crab.
it is.
It's on loan from Andrew Thurber from Oregon State.
All right, shall we mosey along?
Yep, we're going to be going
Yep,
290 and kind of strafing along. I don't think it'll be too significant of a slope, but it'll hopefully be something.
So how tall is Herc?
Open
A little
the
over 2
iris
meters,
back
or
up a bit, Steve. What's that?
How tall's Herc?
how tall is Hercules?
Oh, I don't know. I'm 6 feet and I bump my head on that.
Okay.
I have to stoop to get in under here.
I hit that bubble cam like every day.
We all do, so. Gotta ask Sam, she has all the non-list trivia. We need to get a cheat sheet in here for.
Yeah.
.2 meters tall.
Yeah, that sounds right.
Jumped
Thanks,
over
Meredith.
to the Nautilus Live website where if you click on the images of the ROVs, it will take you to a page with all the information about them for anyone at home who would like to know all of the details.
Zoom in there for a sec, Steve, while I'm getting the belt going. It's good, thanks.
You want to get ahead first before I move, or do you want me to move it?
I think, oh yeah, I need to be on the other side.
Yep.
Okay, zoom out.